Yes, yeah, welcome to yet another episode of solving genetics problems, and I'm talking about what? Oh my god. So I'm going to solve National Examination 2022, and it is genetics now. And as I said, just we had that question in 2021. Now it is the same thing as that one in 2014. And now it is the same thing in 2022. Now in 2022, it was just modified to a certain extent, and someone may not recognize that I'm dealing with the same thing as the one in 2021. So basically, we can go straight to see what was in a question. So now a question was a cross between pure yellow tester and green tester bean seeds produced the yellow tester seeds in F1. You see, so it was a cross between pure. Now we have this pure. This is a very important thing. We had this pure yellow tester pure, and a green tester bean produced the yellow tester in F1. So always when you read such type of question, you must try to generate informations in your head, thinking of what is exactly going on there. So maybe if you don't have any idea about this question, having a cross between yellow tester versus green tester, or the resulting in F1 had yellow tester, you may think that those yellow tester, they are dominant over green tester, because yellow tester, they were seen in F1, and as the principle says, that is the principle of dominance, always it states that a phenotype expressed in F1 hybrids is dominant. So we can think that, oh, probably yellow tester is dominant over green tester. Yes, we can keep on reading. Now on surfing F1 plants, F2 had the phenotypes shown in the table below. So after saving those yellow tester F1 individuals, the resulting F2 had those phenotypes presented in this table. So now we have at least one information that yellow tester, they were seen in F1 generation, so probably they are dominant over green tester. But from those numbers of phenotypes presented in F2, obtained in F2 generation, we can obtain the ratio, because we are provided with the numbers, and always numbers do not provide us with anything. So what we're supposed to do is just to find the ratio from those numbers provided. So if that's the case, we can work the ratio from those numbers. And the idea here is just we have to divide by small number on both groups. We have to divide by small number on both groups. So for example, we have yellow tester, they were in this number 836. Now we have red tester, they had 212. Then green tester, they were 72. So we can arrange those numbers in this way. Now we'll have that 836, the first group. Then we had 1212. Then we had, it was 212, and then we have 78. So the smallest number among those three groups is 78. So we can divide by 78. Then we can divide by 78. And also we can divide by the same number, which is 78. So that's how we're going to work with this. So by 78 will be 1. Now by 78, now here is going to range. Now I'm approximating, but you can work about those numbers those results from this division of dividing this 212 over 78, but it's going to range around 3. So it's going to be 2, will be 2 point something, and when you approximate that, 2 is going to be 3. And if it will be 3, will be 3 point something, not more than 5. Or it is not just 3.5, it is below 3.5. So basically be something like that. And here it's going to be 12. Or if it's going to miss, it will be 11 point something. So when you approximate, it's going to be 12, because even those who call them Mendelian ratios, they were just approximate ratios. So those ratios in genetics, they are not exact number all the time. So sometimes you have to approximate things. So now we obtain this. Now this is a ratio, so we can put this. Now our ratio is 12, 3, then 1. So basically we had this 836 was for yellow tester. Now this was for yellow tester. And then we had red tester, which was 212, red tester. And then we had the green tester, that was 72. So now, always having this ratio gives us information. Now this ratio speaks. So when you have 12 ratio 3 ratio 1, now this indicates that now this was a cross that was involving what we call a simple dominant epistasis. Now this was just a simple dominant epistasis. So if it is a simple dominant epistasis, now it was a cross between heterozygous individuals in those characteristics that show simple dominant epistasis. So from there we have to work for symbols. In a simple dominant epistasis, the one that takes this proportion of 12 is the one that is epistatic to the other, and it will be a dominant allele. And the one that carries this 3 is the hypostatic. So now this will be epistatic, and this will be hypostatic, and then the last one will be for those who call the recessive. Now this will be for recessive alleles. So recessive alleles will be producing this one. So if this 836 was for yellow, so now we can put this caps Y. Now maybe we can let symbols, so you can write there as. So when you are solving this, you will write there as you are letting now those symbols. Now you will let caps Y represent. Now this will represent allele for. Now this will be allele for yellow. Now this will be allele for yellow tester. You can put the tester. Then its recessive, small y. Now this will have that we call the recessive effect, which was green. Now this now small y will represent allele now allele for green tester, and now the same thing, we have got this red tester, which is for this one 212. So this red tester will be dominant, so we'll have this caps R. We let caps R represent allele for red tester. We have red tester, and then its recessive, which is small r. Now this will represent allele, now allele for. Now this will produce the same thing as this one in some cases, which is allele for green. Now will produce the same thing as green tester. Now this will be for green tester in some cases, but in some case your question may state that now this small r has no effect on the color of the tester. So sometimes you can have such cases whereby this one has got no effect. So if that's the concept, now this y, which is caps, will be epistatic for this r. So when caps y, now the idea here is when caps y and d, now anything else, caps r, anything else, when caps y and r appears together, now the phenotype will be yellow. That is the concept. When caps y and r appears together, the phenotype will be yellow. And now we have idea for yellow. And to obtain red, we must have recessive form for both the alleles of gene Y. So when we have this small y, then we have caps G, anything else, maybe caps, another caps G or small g. But when we have recessive form for gene Y, it is when, I mean, now it's not G again. Now this is not G. G was for 2021 now. I have done something which is not right there. So it is R now. So R. So when we have small y, caps R, anything else there, now it is when we will obtain red. So now those are the genotypes now, the possible genotypes. Now if you put those symbols in a Punnett square, we will obtain those things. And then the last thing is when you have small y, caps R, I mean small r. Now this will produce white. This will produce white. So now going straight in our question, we can at least identify those basic things in our question, how the cross was, and put those symbols in a cross. So basically our cross was between pure yellow tester versus green tester. So simply, maybe this one, we can cross in this way. We had, our cross was parental phenotype, our cross was yellow tester versus green tester. But now we have parental genotype, but it has been stated in a question that we had pure yellow. So we have this one for yellow. So pure yellow, it means was homozygous for yellow now. Was homozygous for yellow. So everything was homozygous. And then we have green. We know green is just a recessive, so everything was a recessive for green, so no problem at all. But for this yellow, we must have caps Y, anything else, and then we must have caps R, anything else. So basically, since those F1 individuals, all of them, they were yellow, so this yellow parent should be homozygous as possible, and also it has been stated there that they were pure yellow, so we must put caps Y there, we must put caps R there. And this one is just very simple, something like this, R. So once you have those symbols, you have done. You will form the gametes during meiosis. So once you have done those genotypes, nothing there is going to be complicated. And you are going to finish this question as how I have done for 2021. If you have not watched the video for solution for genetics problem, national examination 2021, just to go and watch those videos. And our question here was just to carry out genetic crosses to show the phenotypes and the genotypes of the F1 and F2. So if the case was to show genotypes and the phenotypes, and it wasn't like that in 2021 that recommended you to use the Punnett square. Here it didn't recommended you to use the Punnett square, so it was just free. You can use genetic crosses in the first cross, then in the second cross, now you can use the Punnett square. So that's how you're supposed to do this question, so you can finish it, just time has gone, so you can just finish it in the same way. But if you have not yet understood how to go with this, you can write in the comment section. Maybe you need me to do the second part of this question to show those crossing, but I think everything has been done. So thank you for your time. If you have any question, write in the comment section, or you can just write through those phone numbers in this channel. And also you can share this video if you think it's valuable is going to help someone else, so you can share it. And yeah, you can subscribe also. That's very basic because I update things several times. So if you have subscribed, you'll be getting those updates. So thank you.